Hello everyone, welcome. So let us start with the process of rDNA technology once we have already known the tools which are required for rDNA technology. I have the tools, I have the restriction enzyme, I have the host cell, I have the plasmid which consists of ORI, which consists of selective markers. I know how will I go ahead selecting the recombinants. All this have been discussed earlier. So now this is the time for the process and the procedure. So the process of recombinant DNA technology, I will have to first isolate the genetic material, which is the DNA, cut the DNA at specific locations using restriction enzyme. Then they have to be amplified. The gene of interest needs to be amplified by PCR. And there is insertion of recombinant DNA into the host cell or organism. Now, first coming to isolation of genetic material, that is DNA. Most eukaryotic cells, whether it is plants, whether it is animals, they carry DNA as the genetic material. To form the rDNA, the DNA should be pure, that is free from all biomolecules. The DNA which is located inside the nucleus, which is enclosed into a plasma membrane, genes intertwined with histone proteins. So you see, whenever I say about I want to go into the gene of interest or I want to go into the gene, then or the DNA, I will have to cross the nuclear membrane. I will have to cross the plasma membrane, again the cell wall in case of plants. And moreover, when I go inside the DNA is already wound around histone. So all these things has to be removed. The cells have to be broken open to release DNA and other macromolecules. So when in the practicals, if we are going ahead, we are going to put some amount of detergents and some amount of salt, and then after that crush the a banana or the plant material. So there's a complete crushing which will rupture the cells and it will remove out the RNA, protein, polysaccharides, and lipids. All this will be removed. So cells are lysed by lytic enzyme like lysozyme, which is bacteria, cellulase, which is plant cell, and chitinase in case of fungus. So all these things. And macromolecules, they are treated with specific enzymes to remove RNA or proteins for protease. So ribonuclease will remove the RNA and protease will remove the proteins. The purified DNA precipitates with addition of ethanol, which appears as a collection of fine threads in suspension. So if you see, a very fine thread will be there and it will be suspended in the alcohol. So you will have to add chilled alcohol, and you should not add it directly but add it to the wall so that it moves inside and we get the DNA intact. Next, coming to the next step, which is cutting of DNA at specific location. So there is a gene of interest. So the purified DNA, the source DNA, desired gene DNA, and vector DNA. So they are being incubated with restriction enzyme for the purpose of digestion at optimal condition. So we add the restriction enzyme to the DNA and we keep it, assuming that restriction enzyme will take the action of cutting the gene of interest and the plasmid. Progressing of restriction enzyme digestion is checked agarose gel electrophoresis, source DNA, and vector. We have already discussed about agarose gel electrophoresis, wherein we will understand based on the size along with the selective marker where the DNA has been cut, which is my recombinant DNA. DNA is negatively charged, so it will move towards positive electrode, and once the gene of interest and cut vector with space of for gene of interest is isolated, DNA ligase is added. This step is the preparation of recombinant DNA, and once I have the recombinant DNA, I will go ahead with PCR, which is polymerase chain reaction. This is a technology to amplify a simple single or a few copies of piece of DNA to generate thousands to millions of copies of a particular DNA sequence in vitro. The components which are responsible will require primers. They are small chemically synthesized oligonucleotides that are complementary to the regions of DNA. We require the nucleotides which is there and the DNA polymerase which is incubated together. There are three basic steps when I talk about PCR. Denaturation, where both the strands on heating they become single strand DNA. Primers are added. They bind to the complementary region. This is known as annealing. Then we have DNA polymerase, the polymerization with nucleotides in the medium genomic DNA act as a template. So they will keep on adding one nucleotide, making the base pairs. Replication is repeated many times. 1 billion copies of desirable DNA amplification and involve thermostable DNA polymerase, Thermus aquaticans, to facilitate replication even at high temperature. Denaturation will take place. So we have talked about denaturation, annealing, extension. These are three basic steps. I need the DNA sample. I need the primers which will attach to the ends. I need the nucleotides ATCG, Taq polymerase that will help in attaching, the mixed buffers, and the PCR tube. Just to give you a broad idea how PCR works. So I have a DNA template. Denaturation is taking place. The DNA is being heated at a minimum of 94 degrees Celsius. This will break the hydrogen bond of DNA template and separate into single strands. Then I will have the primers being added at around 50 to 60 degrees Celsius. The DNA primers will bind, and then there will be extension. The DNA polymerase will go on extending and adding one nucleotides, complementary nucleotides to its 3 prime, because extension always goes ahead from 5 prime to 3 prime end. The next step is insertion of recombinant DNA into the host cell or organism. So once I have my recombinant, they are being already amplified. Now I need to put it into the host cell. So the insertion of rDNA possible by making host competent, which is achieved by chemical treatment, microinjection, bioelastic and gene gun method, or disarming pathogen. We have discussed all these things in the previous video. Phenotype of the organisms alters when rDNA is transformed to the host. For example, the rDNA with gene of interest or gene resistant against antibiotic ampicillin transformed E. coli phenotype, they resistant against ampicillin. So if they are resistant against ampicillin, they are the transformants. We were talking about the screening earlier. When placed on agar plates with ampicillin, the transformed will grow, change in phenotype, and non-transformed will die. Presence of ampicillin resistant gene selected, transformed, and considers as selective markers, as well as I have talked about lactose beta gal gene as well for screening. Obtaining the foreign gene product. See, once I have it into the cell, I get my clones. They are being multiplied, they are being amplified, gene of interest. They are transformed into the host, but the final product, what I am working for, is the final product. So this product, I need to get the desired protein. It can be a vaccine or it can be anything that I need to get. So the alien DNA binds with the cloning vector rDNA transformed to suitable host, bacteria, plant, or animal cell. This rDNA will express itself under the appropriate optimal condition after cloning of rDNA to a particular number of copies. The target protein produced in large scale in heterologous host. If the protein encoding gene is expressed, recombinant protein. Heterologous host, gene or gene fragment that does not naturally present in host and gene expresses itself. So the gene of interest should not be naturally present. Cells which harbor cloned gene of interest grow in laboratory in small scale. Culture of dividing host used to extract desired protein and then purified by different separation techniques. So presently these are the 5 different steps that we have. So the steps, just to put forward once more, we have talked about isolation of genetic material, then cutting at specific sites, selecting of the recombinants, then amplify with PCR, and then insertion of recombinant DNA into the host cell organisms, then the final product, taking up the final product. So this is it in the chapter, and this is it in this particular part, and thank you.